everyone, and welcome to the fourth and final lecture of the GL4U Introduction module set. My name is Amanda Sarabia Butler, and I am the NASA GeneLab Science Lead. It is assumed that if you are listening to this lecture, that you have already listened to the GL4U Introduction Lectures 1, Lectures 2, and Lectures 3, and have also completed the GL4U Introduction hands-on content, including the first 3 Jupyter notebooks, which is an introduction to Jupyter, an introduction to Unix, and an introduction to R. If you have not completed listening to those 3 lectures or completed the hands-on content by finishing those 3 introduction Jupyter notebooks, please go back and do so before continuing to listen to this lecture. Those of you guys who have completed the previous content, welcome to the 4th lecture. This one is going to be an overview of short read sequencing. So just a snapshot of sequencing through the ages here. So the original sequencing was done known as first generation sequencing or Sanger sequencing, in which short pieces of DNA were sequenced and then assembled. And what we're currently at here is next generation sequencing or next-gen sequencing, and this utilizes a technique called pyrosequencing, which we will review in the next slide. This again involves sequencing of short reads or short strands of DNA and then assembling them. And third generation sequencing, which also is happening right now in 2024 when this lecture is being recorded, is single molecule or nanopore sequencing. And for this type of sequencing, it can sequence very long strands of DNA. And so less to know, depending on the length of DNA you're sequencing, assembling is required for this technique. So for the purposes of this introduction module set, we're just going to be reviewing second generation or next gen sequencing because the data that you guys will be analyzing and working with in the omics specific module sets will have come from next generation or pyrosequencing. So just a quick summary on what pyrosequencing is. This is also known as sequencing by synthesis. So in here, representing in red up top is our strand of DNA that we want to sequence. And so for this strand, first, in order to sequence it, we have a sequencing primer represented here in orange. And this is going to be hybridized to the start of our DNA of interest or our fragment that we want to sequence. And just a quick note, although it's not shown here, this priming region, of course, we're going to need to know that sequence. So this was added on to our unknown sequence before the sequencing happened, and we'll get into how that works in later slides. And so we have our sequencing primer here in orange is going to be hybridizing to its complements that is attached to our unknown sequence. And this process of sequencing by synthesis or pyrosequencing is going to be initiated by the addition of one dinucleotide triphosphate, so one ATC or G, at a time. And in this case, we see that cytosine has been introduced here, and then some other things. There are some other enzymes involved in this reaction, including polymerase. So once we have our sequencing primer hybridized, polymerase is recruited, and then it is able to incorporate the cytosine here because the first base that we're sequencing in our unknown sequence happens to be a G. And so that C is incorporated, which releases diphosphate here. Diphosphate is then phosphorylated with the enzyme sulfurylase to generate ATP, known as our energy molecule. And this ATP is utilized by the enzyme luciferase to oxidize its substrate luciferin, which is also part of this reaction. And when luciferin is oxidized, it will produce a light signal. And that was actually hijacked from fireflies. And so once that light signal is produced, we're going to take a picture of it. And so if the next base that we're sequencing is complementary to the known dinucleotide triphosphate that we added, in this case cytosine, it will be incorporated into this growing strand, and it'll give off a light signal through this reaction that we'll be able to take a picture of. So if there was not a G here next, we would not see a light signal because, of course, C will only hybridize with guanines with G. And then another enzyme, a pyrase, will be added to this reaction, and a pyrase is essentially going to eat up and get rid of all of those loose cytosines in this example, and we'll be able to wash this away and then start all over again with a different dinucleotide triphosphate. And this will continue, and we will see those graphs generated here, which we then can, this is known as a pyrogram, and we then can read to see the sequence of our unknown DNA template. So in this example here with pyrosequencing, we're introducing just one nucleotide at a time. However, this technique has gotten much more sophisticated, and now sequencing by synthesis is done by adding all 4 of your nucleotides at a time, so your A, T, Cs, and Gs, and each one of them are attached to a different color fluorophore. And so that way we will know with each cycle or each base of our unknown that's being sequenced, we will always have either the A, T, C, or G incorporated, depending on which of its complement is there. And the light signal that's released will be a specific color, and that's how we will know if it is an A, T, C, or G that was incorporated in that growing strand. So in the Illumina sequencing workflow, that is the workflow that we're going to go over here, I'm going to walk you guys through how we prepare samples for sequencing. And the reason I'm talking about Illumina is because Illumina is a very successful biotech company that specializes in next generation sequencing technology that ultimately utilizes this pyrosequencing method. And still to this day, as of the end of 2024 here, roughly 90% of all sequencing worldwide is performed on an Illumina instrument. And in fact, in GeneLab, we have a sample processing laboratory where we also have an Illumina instrument to perform our in-house sequencing. And so I'm going to be going over the Illumina workflow. In all of these slides, resources are going to be found at the bottom of the slide with links to learn more and for where I pulled and got the information from. So the Illumina sequencing workflow essentially has 3 main steps. The first being library construction, and then cluster formation, followed by sequencing by synthesis. So because our omics-specific module sets will include sequencing of either DNA or RNA, depending on which omics you're going to be learning about, in this introduction slide deck here, I'm going to talk about how we prepare libraries for both DNA sequencing as well as RNA sequencing. So starting with DNA sequencing first here. The first step is going to be to create DNA fragments with some kind of means to attach adapters from the extracted sample DNA. So if you remember in the pyrosequencing, we had our unknown region, but then we had to have that known region at the beginning in order to create primers that will hybridize to it in order to initiate the sequencing of the unknown. So we have to be able to attach a known sequence to our unknown DNA that we want to sequence. And so here, this is going to start first. You're going to isolate DNA from your samples of interest. There's a variety of different ways you can perform DNA isolation. There's different types of kits. You can also do different types of chemicals and chemical washings to isolate your DNA. So once you have your DNA isolated from your samples, you want to do 2 things in this first step here. One is you want to chop it up into smaller pieces. So isolated DNA can be very, very long, but for short read sequencing, as the name implies, we are only going to be sequencing short pieces of DNA. So we want to make sure that we chop up the DNA that we isolate And then also here, as I mentioned, we need to have some way to attach a known sequence onto our unknown DNA target here. And so I'm going to go over just 3 methods for how this is done. There are many more methods for how this is done in different library prep kits that can be used, just going over 3 of them here. This first one is Illumina's DNA prep, which was formerly known as their Nextera Flex kit. And in this technology, it uses something called tagmentation. And so in here, there are beads that have these little transposase enzymes, and transposases are enzymes that will interact with DNA, introducing random double-strand cuts. So it'll cut randomly throughout the DNA strand and cut both pieces of DNA, so both your sense and your antisense. And then also within these little complexes on this bead are short oligos, which are going to be the priming sequence that we're going to be adding on to our DNA fragments. So here in this technology, we are going to both cut the DNA into smaller pieces while simultaneously adding a sequencing primer onto either end of our cut fragments. So that's what you see here, our unknown in gray, and then our priming sequences on both the 5' and the 3' end in blue and green, respectively. Another means to perform this first step is to go ahead and take your extracted DNA from your sample and then to chop it up. You could chop it up manually in this case by putting it essentially in a small blender and chop it up into different pieces. Or you can do something like adding a divalent cation, which will allow cleavage at random locations throughout your DNA strand, making smaller pieces. And then once you have these shorter pieces of DNA here, what's happening is we are utilizing something that we have in our own cells that adds adenosine nucleotides to the 3' end of blunt fragments. It's an enzyme called poly-A polymerase because it will add adenosines to that 3' end. And so here we just add 1 adenosine to the 3' end of either strand in our double strand DNA fragments. And that's going to come important as we'll see in the next slide. A third mechanism to add in known sequences to be able to sequence our unknown regions. This example is from the Amplicon library preparation kit of Illumina. And Amplicon sequencing is a way to try to identify unknown organisms in a sample that has multiple different organisms. So in here, you take your sample, such as soil, you isolate all the DNA from that sample, and then in this case, the amplicon that we're amplifying is a variable region of the gene that encodes 16S ribosomal RNA. And what you want here is flanking that variable region, a conserved region that we know the sequence to, so that way we can create primers that will hybridize to that conserved region or known sequence, flanking our unknown region that we want to amplify. And this is the region we ultimately want to sequence from our sample. And then it'll also add in this spacer. A spacer, again, is just a string of A, T, C's, and G's. And then lastly here, attaching on to that spacer sequence is going to be the sequencing primer sequence. So similar to the sequencing primers that were added on up at the top. And then this is going to undergo PCR amplification using these primers. For those of you guys who are unfamiliar with PCR amplification, it's essentially making copy of DNA, and you're only going to copy the region that's going to be in between your 2 priming regions, and it will add on anything that you have flanking your primers. So in this case, our sequencing primers along with this spacer region. And so at the end of this step for the Illumina Amplicon method here, we're going to end up with this region in orange, our unknown variable region that we want to sequence, the conserved region that's flanking either end of that unknown region, a spacer, and then these sequencing primers on the end of that, which are ultimately going to be used in the sequencing by synthesis method. So at the end here, in these three different ways that I showed you, again, there are more ways in this, more different types of library prep kits on this. I'm just going over three common ones. And here at the end, we have our unknown region that we want to sequence, and then we have some means on either end of these DNA fragments to add on the rest of our adapters, which I'll get into next. So next here is attaching the adapters. So sequencing adapters have three key regions in them. One is going to be the sequencing primer, which again is just a known strand of A, T, Cs, and Gs that we're going to use to be able to prime it for sequencing by synthesis, similar to what I showed you in the pyrosequencing overview. So the sequencing priming region is one of the three parts of the adapter. The next is the index. The index, again, is just a string of known A, T, Cs, and Gs. These indices are going to be unique to each sample. So any target DNA coming from the same sample will have the same index. This will allow us, after sequencing, in which we will combine sequences from multiple different samples together to do our sequencing. So in order to figure out which sequence each sample came from, we will be using this index part of the adapter to do that. So again, your index is going to be unique to all sequences that came from the same sample. So your index corresponding to your sample. And then lastly, here we have these P5 and P7 regions. Again, just a strand of known DNA sequence. And these regions here are going to be complementary to the flow cell, which is the platform that is used in Illumina instruments to hybridize this library on to be able to sequence that known region, as we'll see in later slides. So in this step here, we want to start with whatever we ended up with in the first step and make sure that we add on whatever was missing from our three critical pieces of our adapter. So again, looking at the three methods that we started with in the previous slide. First here is looking at the Illumina DNA prep method. If you recall from the previous slide, that first step after tagmentation, we have our short pieces of target DNA or template DNA that we want to sequence. And then we added on these known sequencing primer regions, right? So that's these regions seen here. And so in this step, we take advantage of these known sequencing primer regions to create primers that also include flanking it, both an index sequence on either side if you're doing dual indexes, and this P5 or P7 sequence, which again will allow us to bind to the flow cell in order to sequence our unknown DNA template. So then we're going to undergo PCR amplification, which is just making copies of this double-stranded piece of DNA, but now each copy will also include this index region and this P5 and P7 region on either strand. And so after this step, we're going to be left with our unknown DNA target here, a template that we want to sequence. We're going to have our sequencing priming regions on either side of our unknown strand. Our indexes, which again, your index is going to be specific to your sample. So every piece of DNA that came from the same sample will have the same index. And then lastly, flanking this is going to be your P5 and your P7 sequences, which are going to allow you to attach to Illumina's flow cell to enable sequencing, which we will see later. Looking at the A-tailing method, I should mention this is a library prep kit created by the company IDT here that I'm pulling this from. And so in here, if you remember in the previous slide, we had chopped up our DNA either manually or with some kind of random enzyme or divalent cation to create DNA fragments, then utilize poly-A polymerase to add adenosines to the 3' end of these DNA fragments. That 3' adenosine created a sticky end DNA does not like to be unpaired, and so we take advantage of those A tails that we have on our 3' ends of our double-stranded DNA and use an enzyme called ligase to ligate on our adapters. And so here our adapters are going to be the priming region on either end. The index, as you see here, is I5 and I7 on either end, and then our P5 and our P7 regions to be able to attach to the flow cell. And you'll notice at the end here, let's start here, of these adapters, we have a hanging T or thymine, which is going to hybridize to its adenosine that was hanging on the fragments from the previous step. And so ligase will come on and allow these two to bind here. And now our unknown fragments will have those 3 critical pieces needed for sequencing. Again, those are going to be the sequence priming region. The index will help us identify which sample the sequence came from, and then our P5 and our P7 regions, which will allow us to attach to the flow cell to sequence. And then lastly here, this last example I'm going over utilizes Illumina's Amplicon library preparation. And if you recall from the previous step, we had our variable region that we will be sequencing along with the flanking conserved region that will also be sequencing, a little spacer region here, followed by our sequencing priming regions. So again, similar to the DNA prep here, we're going to take advantage of those known sequencing priming regions to create primers that also have an index as well as a P5 or a P7 region flanking that sequencing primer. And then we're going to undergo PCR, which again, just making copies of this. And this time it will add on these 2 additional pieces of the adapter. So here in our final library here, we will have, once again, our priming sequencing regions in yellow and light blue here, our indexes on either side in red and in blue here, and also our P5 and P7 regions, which will allow us to attach to the flow cell in orange here or in pink. And so the very last step of DNA library preparation is going to be to undergo a few cycles of PCR amplification. So this is where we take our final libraries, which again has those 3 critical pieces of our adapters attached on, sequencing priming region, our index to identify our samples, our P5 and P7 to attach to the flow cell. And we are just going to amplify these final libraries to ensure that we have enough material to put on the sequencer. And so that same PCR step is going to happen for each one of these different methods. So the next thing that we want to do after we have our libraries ready to go is we first want to do a quality control check on our libraries to make sure that they are ready to be sequenced and they look the way we want them to look. I should also mention throughout the library preparation, you'll notice in each step we were adding some things like our extra pieces of our adapters to be added on, sequencing primers, etc. So after each step, it critically undergoes a wash step to remove any of that remaining adapters that didn't get added on to our unknown DNA template. And it's important that any additional remaining adapters get removed because they will be problematic. So doing this next step, which is performing a quality control check on our libraries, is going to be critical to make sure that these are nice and clean libraries and ready for sequencing. So how we perform our quality control checks on our libraries is essentially going to be utilizing electrophoresis. And for those of you guys who are unfamiliar with electrophoresis, we take advantage of the fact that DNA is positively charged because of its phosphate backbone. And what we do is create a polyacrylamide gel and then put that gel in a buffer and put a current through that buffer such that the positive current is going to be on our positive electrode is going to be on the top here, where we're going to load our samples on the polyacrylamide gel. Excuse me, the negative electrode is going to be on the top here, where we're going to load our libraries into the polyacrylamide gel. And on the bottom here is where we're going to have our positive electrode. So again, negative electrode on top, positive electrode on the bottom. And then we're going to go ahead and load our DNA samples, our library samples in our case, into these wells. And importantly, one well, at least we're going to have a sample that contains DNA that has pieces of known sizes. So we know which sizes the fragments are in our controller, in our ladder here. And then we're going to go ahead, load our samples in our ladder, run this current through the gel, allowing the DNA fragments to separate out. And these DNA fragments will separate out such that smaller pieces of DNA are going to travel through this polyacrylamide gel faster than larger pieces of DNA because they're going to get hung up and not be able to go through those pores as easily. And then we will stop the current and take a look at these bands. So these bands are going to represent different sizes of DNA that are present in our sample. And we have known sizes of DNA in our ladder. And the thickness of these bands are going to correspond to how much of that size DNA is in our sample. So again, smaller pieces of DNA are going to be on the bottom, larger pieces of DNA are going to be on the top. And we're going to be able to utilize this to see what the size of our libraries are. And this information can then be plotted here on what's known as an electropherogram, where we take advantage of knowing the size of our markers, of our ladders, to create a size map on the x-axis here. And then on the y-axis, we have sample intensity. So again, looking at the thickness of the bands at each size location on that gel. This here, we're just seeing an example of our markers at the lower end at 25 base pairs here, at the upper end around 1500 base pairs here. And then we see our unknown sample of interest has a range, right? So we're going to see a few different sizes of our libraries in this case, all roughly around 185, 200 base pairs, it's looking like here. So these electropherograms derived from a gel electrophoresis is what we are going to be using to perform a quality check on our libraries that we just created. And so, although I showed you what it looks like kind of on the macro scale here, that gel electrophoresis has really been minimized into a couple of different types of instruments. More common ones used are known as a bioanalyzer or a tape station, and it makes this gel electrophoresis very easy, where you just load your sample, put it in the machine, and the machine will spit back out to you that electropherogram to help you assess the different sizes that you have in your library samples. And so this here is showing what a good library looks like. So this is what we're going to expect from our libraries. We should see our low end marker as well as our high end marker that was used to determine the size. And then also we should see one single peak. Usually, it's going to be spread. It's not going to be very sharp peaks, so kind of broad. And this would be the range of the sizes of our libraries for our sample. And so as you can see here, this is a question I'll have before, but you can go based on the sizes on the x-axis and see where the majority of your sample, what size is the majority of your sample, the majority of your libraries. And we want to make sure that this size here is conducive with how long we're going to be sequencing on the sequencing instrument. And this is an example of what a bad library looks like. So as you can see, we have both our lower and our upper markers here. However, we don't just have one nice clean peak at the size that we would expect our libraries to be. Instead, we have our sample peak here, but we also have what's known as a primer dimer. If you remember, after I got done talking about library preparation, I mentioned that there are washes done in between, and that's to remove any additional primers that might still be in there with our libraries that haven't attached to our unknown DNA template. And why it's so critical to remove these is because if you don't, either side of that primer will just attach to itself and not have any of your unknown sequence in it. So it would just be the 3' adapter and the 5' adapter attached to each other. And this is going to be a huge issue when we go to sequence our libraries, as we'll see in a minute. So very important that we make sure that we do not see any primer dimers when we do our quality check on our libraries for each sample. The other problematic things in this example of a bad library here is we also have this other little peak here at around, looks like around 75 base pairs. That shouldn't be the size of our libraries because we chopped up our libraries at a specific size range. And again, that depends on how you want to sequence your samples. And so we should not see any of these low end artifacts. And we also see an artifact at the very high end that goes beyond our upper range. So this shows us that when we did our filtering steps during library preparation to size filter, size select our libraries, we saw some larger pieces of DNA come through, as well as some smaller ones. And we also did not successfully remove primer dimers here. So if you see this, you do not want to sequence this library. So when assessing your library quality, you want to look for the following. You want to make sure that you have no adapter dimers like you see here. And why? That is because if you sequence your sample and it has adapter dimers in it, because those adapter dimers are small, they're going to be roughly around 125 to 145 base pairs, depending on the length of those 3 different pieces of your adapter. So that means they're going to be small. They're not going to contain any of your unknown DNA that you want to sequence, but they are going to contain everything that they need to be sequenced. They're going to have your sequencing primer. They're going to have your P5 and P7 to attach to the flow cell. And because they're smaller than your actual libraries here, the flow cells have a bias towards short pieces of DNA. So those primer dimers are going to be more likely to latch onto your flow cell and get sequenced than the library yourself, because the libraries are going to be larger. And we'll see just how much of an issue that is in the upcoming slide. You also want to check your library size and make sure it's consistent with the number of desired sequencing cycles. So throughout that library preparation, with your final libraries, you're going to end up doing a size selection, and you want to make sure that the size of your libraries and ultimately the size of the fragments that you started with are going to be dependent on the length of your sequences. So for example, if you ultimately want to sequence your libraries at paired end 250, that means you are going to be sequencing your unknown fragment at 250 base pairs from the 5' end and 250 base pairs from the 3' end. And you usually want to have a little bit of overlap between the sequences that are generated from your forward and your reverse reads here. And so if you're sequencing 250 base pairs in this direction and 250 base pairs in this direction, and you want some overlap, you're going to want your fragments that you start with to be somewhere between 450, 475 base pairs. Again, that way the fragment that you're sequencing, if it's 450 base pairs and we sequence 250 in this direction, 250 in this direction, so 500 total, about 50 base pairs from your forward and your reverse read will overlap there. So you'll be able to reconstruct the fragment that you sequenced. In addition to the size of your fragments, when you're assessing the size of your library, you also have to remember that when you're assessing your library sizes, to that fragment, you have added on your adapters. And depending again on the size of your adapters, and that'll be dependent on the length of your index. The sequencing priming length is pretty consistent because it's based on the sequencing primers used in Illumina's flow cell technology, as well as your P5 and P7. And so the size of your adapters are going to be roughly 65 base pairs each. So you have one on each side. So it's going to be roughly 130 base pairs. And so when you're looking at the size of your library, it should be the size of your insert or template DNA that you want to sequence, plus the size of your two adapters. And so if you're sequencing at paired end 250, a good library size is going to be around 600 base pairs. This is assuming that adapters are about 65 base pairs each, so 130 base pairs total, right? Because you have an adapter on each side. And that your insert length is around 470 base pairs. And so if it's 470 base pairs and you're sequencing a total of 500 bases, 250 this side, 250 that side, you'll have roughly a 30 base pair overlap. And so with an insert length of 470, an adapter length total of 130, that's what gives me that roughly 600 base pair library size. And so just taking a look at the library sizes of both of these libraries, on this side, it's around 225 to 675 base pairs. So that's pretty good. I probably want this to be a little bit larger than that. So we're going to get some redundancy, some extra redundancy in our read 1 and read 2, our forward and reverse read sequencing. But that's still a pretty good library. Again, this library is very clean. So I would move forward with this one. And with this one, even just the library sizes here, we're looking at around 200 or so starting here at the library. And then if this artifact is part of the library too, over 4,000 base pairs, that's just a crazy range. And again, this library is definitely going to need some additional cleanup or maybe just to even restart and build the library from scratch. So again, assessing library quality, we do this with electrophoresis. Electrophoresis has been miniaturized into instruments known as bioanalyzers or tape stations. And when we're doing this assessment, we want to look for adapter dimers and the size of our fragments to make sure that adapter dimers were removed and our fragment sizes are consistent with the amount of sequencing, the length of the sequencing that we're planning to do for our fragments. We also, with our quality control, want to determine the library quantity, right? So we're going to want to know how much library we have, so that way we make sure that we load the appropriate amount on our sequencer. And you can do this with a variety of different instruments. One way to do it is with an instrument called Qubit. Another way is you could do quantitative PCR to determine how much libraries you have. And so just to reiterate with these adapter dimers, so I know I described this, but this is what they look like here. So this is when you have your two adapters, but instead of latching on to either side of your DNA template that you want to sequence, instead they just latch onto each other. And if they're not filtered out in your filtering steps during library preparation, this is what you're going to be left with in your samples here. In the examples I was showing previously, it was dual index adapters. In this, we have a single index, so just an index on one side. Both exist. I prefer dual index, gives you more confidence in identifying your samples if there's an index on both sides. But here, we just have it on one side. So anyways, you see here that our two sequencing priming regions have come together without any template in between. Also have your index and your P5 and P7, which will allow this to attach onto the flow cell. This will allow sequencing by synthesis to occur. And when this fragment is sequenced, all that's being sequenced are your adapter regions, not your unknown DNA template that you actually want to sequence. And why this is such a problem is, again, looking at a nice clean library here with 0% adapter dimer. Here, this is what a library looks like, this peak being roughly 5% of your sample making up adapter dimer. And because of the bias of the flow cell being more likely for smaller pieces to latch on, even with as little as 5% adapter dimer, it's going to end up being roughly 60% of your sequences coming off the sequencer. So just here, again, this comes from Illumina's documentation where they did an assessment of the effects of primer dimers on sequencing. And so if you have 1% primer dimer, it ends up coming out to roughly 6.5, 7% of your sequences, which isn't great, but it's okay. So usually at GeneLab, we make sure that our adapter dimers, if they exist at all, it's less than 1% of our libraries. Because once you get up to 5%, so just 5% of your libraries, if they are adapter dimers, it will take up 60% of your reads. That is a lot of real estate on the flow cell. So essentially you are throwing away 60% of your data. If you get up to 10% adapter dimer, you are looking at about 85% of your reads just being reads of adapters, which means that you only have roughly 15% of sequences from your target region. And so you are throwing away 85% of your data. So again, I can't stress enough how important it is to take a look at the quality of your libraries before you move on to sequencing. Moving on next to what library preparation looks like for RNA samples. So here first, after we isolate our RNA from our samples, we're going to have to remove the ribosomal RNA that is in there. The reason for that is because ribosomal RNA takes up the majority of the RNA in our samples. Ribosomal RNA is very important. It is a molecule that is critical to creating the large and small subunits of the ribosome, which are essential to be able to translate RNA into protein. So without our ribosomes, we can't make protein, and it's critical to make protein in order to do all of the things that living organisms do. And so it's very essential ribosomal RNA. However, it's super highly conserved across organisms. It's not very interesting usually to look at unless you are specifically looking at ribosomal RNA. You really want to make sure you remove it, or else the majority of what you're going to be sequencing is going to be ribosomal RNA. And so here we're just looking at a graph that I got from NEB that's just showing in a RNA sample. This is a human reference. It's looking like roughly 91% of the RNA that was isolated here is ribosomal RNA, leaving less than 10% for the RNA that might be interesting to us. And just as a reminder here, before I get into the different ways to remove ribosomal RNA, for those of you guys unfamiliar with RNA or who need a refresher, so when we make RNA that we ultimately want to translate into protein, it is called messenger RNA. And before mature messenger RNA is created, we first transcribe RNA or pre-mRNA from a DNA template. And that has these different regions in it. So it will have a 5' untranslated region. It will have several exons shown here in red, as well as introns in between those exons shown here in a grayish blue, and then also a 3' untranslated region. This pre-mRNA is going to undergo RNA splicing, in which the introns are going to be spliced out, the exons are going to be brought together here, and then some additional things are going to happen. Because single-stranded RNA is very unstable, in order to help stabilize it at least long enough for it to get translated into protein, what happens is a 5' cap is added onto the 5' end to protect the 5' end from degradation. And at the 3' end, a poly-A tail, which is just a string of adenosines, is added on to protect that 3' end from degradation. And so in order to remove ribosomal RNA, one way we can do it is if we're only interested in looking at the messenger RNA in our sample, we could take advantage of the fact that mature messenger RNA has a poly-A tail, and we can design sequences, synthetic sequences that are polythymines, so poly-DTs attached to a magnetic bead. and then introduce that into our samples so that these poly A tails will hybridize to that poly DT that we added. And because we latched that onto a little magnetic bead, we can use a magnet to then pull out our messenger RNA from our sample and then only carry that messenger RNA into the subsequent library preparation steps. So by doing that, we're positively isolating messenger RNA by selecting that poly A tail. So that's one way we could do it where we shouldn't have any ribosomal RNA in there, or at least very little. We'll get into why in a minute. But the majority of our sample in this case should be RNAs that have polyase attached to it, so messenger RNA. Some reasons why we might not want to take this approach and want to take a different approach is if your samples are already pretty degraded, that poly A tail might not be there on all of your messenger RNA. And so you'll only be able to see RNAs that have an intact poly A tail. So again, if you're working with degraded samples, this probably isn't going to be the best route. Also, if you're interested in non-coding RNAs, so long non-coding RNAs, microRNAs, no RNAs, pRNAs, your favorite type of non-coding RNA, you're not going to be able to capture it using this method, right? So if you want to look at other RNAs in addition to messenger RNA, what you can do is target the ribosomal RNA to remove that from your sample. And so in this case, you can use an enzyme that targets and degrades ribosomal RNA selectively. You can also use the magnetic bead mechanism again, except this time attach the sequence that's going to be complementary to ribosomal RNA. So that way the ribosomal RNA in your sample will attach on, and then you could pull it down with a magnet. Those are just some ways that you can remove ribosomal RNA from your sample. And then what you're left with, hopefully, is a sample free of ribosomal RNA. So just looking at what your RNAs look like in either of these methods, this first is looking at that poly A enriched, or when we use a poly DT to select for RNAs that have a poly A tail, we'll see that we now have the majority of our remaining sample is going to have exonic regions indicative of our messenger RNA. I do want to point out here that you don't only have these exonic regions. You also still have some intronic regions, even some intergenic regions, and sometimes even some ribosomal RNAs that sneak in there. The reason for that is because when you use this mechanism of putting in a poly DT attached to a magnetic bead, it's not only going to be hybridizing to poly A tails on messenger RNA. If there are RNAs, other types of RNAs in there that have strings of A's as well, those are also going to hybridize to that poly DT that you introduce to your sample. And so it's not particularly selective to just messenger RNA. It's selective to RNAs that have long strands of polyadenylation. And so that's why you'll get some of these other types of RNAs in there as well, although the majority of the RNA that you isolate, again, as long as your sample is not degraded, should be messenger RNA. And then just taking a look at what this RNA breakdown looks like if you use the ribodepletion method. So again, in this method, you are going to select and remove ribosomal RNA from your sample, leaving all the other RNAs in your sample. And so here you'll see more so of these intronic and intergenic RNA, and as well as your messenger RNA, your exonic RNA here. Because all that you did was remove ribosomal RNA. So all the other types of RNA should be left in your sample. You will also see some ribosomal RNA reside in here. Ribosomal RNA, removing it is almost never 100% efficient. So you will probably get some trace amounts of ribosomal RNA. Okay, so once you've isolated your RNA from your samples and you found some means to remove ribosomal RNA contamination, either by removing ribosomal RNA or positively selecting for message RNA, you are now ready to take that into library preparation. So I'm going to walk through the Illumina TrueSeq stranded total RNA library prep kit method. This is the one that we use at GeneLab. Again, there are other kits and other methods to generate libraries from RNA. This is a common one that's used, one we use in GeneLab. So this is the one that I'm going to walk through. As a reminder here, what's being shown in this first step is what I just talked about in the previous slide. So you start with your isolated RNA, and then you get rid of your ribosomal RNA. So here we are taking our isolated RNA and removing ribosomal RNA through ribosomal RNA depletion method. And so we're left with all other RNAs in this example. And also importantly, we are going to have to chop up our RNA. This is again because we are doing short read sequencing. And so we want to make sure that our RNAs are in the fragment size that we're ultimately going to want to sequence on the sequencer. And you can chop up your RNA either with an enzyme, ideally a non-specific enzyme. You can manually chop up your RNA through like a very small blender. You can also introduce divalent cations, which will bust up your phosphate backbone in random locations to chop up your RNA. I believe this kit uses the divalent cation method. So now, after this step, our ribosomal RNA has been removed. We have short pieces of RNA left that we want to sequence. And so we're going to go ahead now and synthesize our first strand. Remember, we are starting with messenger RNA. Short read sequencers like Illumina are only going to be sequencing DNA. And so we want to first convert our RNA into complementary DNA or cDNA. We do this using the enzyme reverse transcriptase, which we hijack from viruses, I believe, to be able to use this RNA template that was derived from our sample to generate a complementary DNA strand. Keep in mind here, although we do see a sequence here, at this point, we do not know what this sequence is. And so we go ahead, use reverse transcriptase. It is using a random hexamer, so just a random sequence of 6 different nucleotides, and it's supposed to be random because it's supposed to be sequencing at random locations within your message RNA, or within, sorry, your total RNA here, to make sure that you transcribe all of the RNA in your sample. And so again, using reverse transcriptase, we make our complementary DNA from our initial messenger RNA that came from, or RNA that came from our sample. Could be messenger, could be other types of non-coding RNAs. Next step here is synthesizing our second strand of our cDNA. So now using this first strand that we created in this step here, we're going to use that as a template now to make a copy of it, our second strand of cDNA. However, importantly here, we are incorporating, instead of thymines, we're incorporating urisols into the second strand. This will allow us to preserve the orientation the RNA started with. This is important because RNA can be derived from either the sense or the antisense strand of our DNA template. And so in order to make sure that we keep the orientation that was in our original sample, we have to make sure that we create what's known as a stranded library here. So this is how that's done in this Illumina kit, is when that second strand of cDNA is synthesized, it is done such that urisols are introduced here instead of thymines, and we'll see how that comes into play later. The next step that we want to do, again, this is an unknown sequence at this point, and so we want to make sure that we have a means to add adapters of known sequences to be able to sequence this. And so here, similar to what we saw in the DNA library prep kit, we use poly-A polymerase to add adenosines to the 3' blunt ends of our cDNA strands here. And now we have this A overhang that will create a sticky end that we're going to take advantage of to add adapters. So our next slide here, we are going to be adding adapters. Our adapters are looking like this. So we'll see that in this example, we have a single index adapter. So the index is only on one side. And so here we see our sequencing primers on our 5' side and our other sequencing primer here on our 3' side. Just this side has the index, and then both will have our P5 and P7. As a reminder, the sequencing priming regions are designed to be able to latch onto the primer to perform the sequencing in our sequencing instrument. Our indexes are so that way we can identify which sample each sequence came from. And our P5 and our P7 regions are going to allow us to attach onto the flow cell. And then you'll notice here that we have this T overhang. This T overhang was added so that way it can bind to its complement adenosine, these A overhangs that were added on in the previous step. And this is done using the enzyme ligase to add on these adapters to these cDNA fragments. And so at the end of this ligation step, we should have our unknown cDNA sequence here, followed by our sequencing primers on either side, followed by our index on one or both sides, and then lastly, our P5 and P7 regions to attach onto the flow cell. So as I mentioned here, this is a single index adapter because we just have an index on one side. This is just another example from IDT of what a single index looks like. Your insert here is going to be your unknown cDNA that you want to sequence, sequencing primers on either side. Because it's a single index, we have a single index on one side, and then our P5 and P7 regions to attach to the flow cell. For unique dual index, it would look like this. So we have our insert sequencing primers on either side, then an index on both our I5 and our P7 side, followed by the P5 and P7 regions. There's another type of index, and this is actually the one that we use at GeneLab that we can have here. And for this one, we again have our insert and our sequencing primers flanking our insert. We also have a dual index, so an index on the P5 side shown here as I5 index, an index on the I7, on the P7 side shown here as an I7 for the index. However, in here, we also have an additional strand of ATCs and Gs known as a UMI, or a unique molecular identifier. And this sequence is going to be unique to our insert, so to our sequence. So that means that every sequence, in this case, from the same sample, will have the same index, but every sequence in that sample is going to have a unique UMI, right? A unique strand here. So this is going to help us identify technical duplicates when we go to the next step, which is our PCR step. And we'll see why technical duplicates can be a problem when you guys take the RNA-seq omics module set. So just importantly, this UMI is added before the final PCR step, and it's going to be unique to each sequence. And so at this point, we are ready for our final PCR step. And this is where that second strand cDNA that has the uracils in there come into play. Because when we do our final PCR step to amplify our libraries before we go on to QCing and then sequencing, we add a polymerase in this PCR step that is not able to incorporate past a uracil. And why this is important is this means that only one strand, only one orientation here is going to get amplified. And this is how it allows us to preserve the orientation of our original RNA that came from our sample. So if we didn't do this, both of these strands would be amplified in both orientations, and we would have no way of telling what the original orientation was. So because that second strand has uracils in it, and then in this final PCR step, we do not amplify that strand. We just amplify the orientation of the first cDNA strand. We are able to preserve the orientation of our original RNA from our sample. And so now after this PCR step, we have our final libraries here with our insert cDNA that we're going to sequence, our sequencing primers on either side, our index either on one side as shown here or on both sides, and then lastly our P5 and our P7 region to allow us to attach on to the flow cell. Next is going to be RNA library QC. So similar to what we did with our DNA libraries, we are also going to want to QC our RNA libraries to make sure that they are nice and clean and the right size before we move on. As a quick reminder, this again utilizes electrophoresis. Electrophoresis involves a polyacrylamide gel that's inside of a buffer that has a current that goes from a negative electrode on top where we load our samples to a positive electrode on the bottom. And that way, when we load our samples at the top here, we take advantage of the fact that DNA is negatively charged because of its phosphate backbone, and the DNA will flow from that negative electrode towards the positive electrode, and smaller pieces of DNA are going to be able to travel faster than larger pieces, which are going to get hung up as it's traveling through that polyacrylamide gel. And we also have a marker that we're running as well. This marker has known sizes of DNA, so that way we know which size each one of these bands represent, that we can then use to build a ladder of specific sizes to map our unknown samples to. And lastly, the thickness of these bands are going to represent the amount of DNA that is that size. And this again is going to be plotted on what's known as electropherogram. This electropherogram is actually looking a little different. This is the time that each peak is coming off and then the fluorescence measured. This is just showing one of RNA. This RNA still has its ribosomal RNA in it, as we can see by the 5S, 18S, and 26S ribosomal fragments. But the final library will, of course, have had that ribosomal RNA removed and undergo size selection to be the size that we want based on the length that we're sequencing. And so what we see here in this example of an electropherogram of our RNA library here, again, we see our heavy marker here up at the top that's going to be represented by this peak here in our electropherogram. We see our lightweight marker here at the bottom, which is going to be represented by this in the electropherogram. We see this nice thick spread of DNA here, cDNA in our case. This is going to represent all the different sizes of our library, which is shown here in this peak. However, this band here that we see is slightly smaller than our main library. We see it right here, looking roughly or hovering around that 140 base pair. That is what it looks like when you have adapter dimers again. And remember, adapter dimers are going to be a problem because even a small amount of adapter dimer contamination is going to take up a lot of real estate on our sequencing flow cell. So we're going to go through this again. Just to reiterate, libraries are evaluated using a bioanalyzer or a tape station. These are essentially miniaturized electrophoresis shells that will generate electropherograms for us to assess the quality of our libraries. This is just looking at a couple examples again of good RNA-seq libraries. So we have our low-end marker in both of these, our high-end marker, and then we see just one clear peak, which represents our libraries. And these, of course, have a little bit of a spread because you're always going to get a range of sizes for your libraries when you do your size selections here. Just to remind you again, this is what a bad library looks like. Here we have quite a bit of adapter dimers, very little of our actual sample library. This is going to be a huge problem when we go to sequence. So again, when assessing library quality, similar to DNA libraries, for RNA libraries, we're also looking for the same thing. No adapter dimers. Why? Because even a small amount of adapter dimers would take up a lot of our sequencing reads on the sequencer. We want to make sure that our library size is consistent with the number of the desired sequencing cycles or the length of our sequences that we're going to be doing. And just as a reminder on how you calculate that is if you're sequencing at paired end 100, and you're thinking about what library size you want. So paired end 100 means that you are sequencing your unknown cDNA insert in 100 base pairs in this direction and 100 base pairs in this direction. So 200 base pairs total. So you want your insert size, your cDNA size, to be just under 200 base pairs. So you have some overlap. So it could be roughly 170 base pairs there. But you also want to consider the size of your adapters when looking at your library size. So 170 base pairs for your insert length, again, because you're sequencing 100 in this direction, 100 in this direction, 200 total. You want some overlap between your forward and your reverse reads so you can reconstruct your insert. And then also the size of your adapters, if they're roughly 130, you're looking at, you want a library roughly 300 base pairs long. So again, showing here, assuming adapters are around 65 base pairs each or 130 base pairs total, you want to see that your library size is roughly 300 base pairs. That would give you an insert length or your cDNA length of around 170, allowing about a 30 base pair overlap between your forward and your reverse reads. And then just taking a look here, what is the size range of libraries 1 and 2? Library 1 is looking around, it's around 175 to around 250 base pairs. So if we're using these metrics, I would say that this is a little small. So you're probably going to get a bit more overlap between your forward and your reverse reads. That's okay, although, I would probably like to see this a little bit larger. And then library 2 is actually looking a little better for me because we see that there's just some larger sizes here. So this one between around 150 and 275 base pairs, so a little bit closer to that 300 that we would be looking for here. And just to reiterate, the damage of these adapter dimers, also in addition to looking for adapter dimers and fragment size, remember you also want to quantitate your library so you know how much to load on your flow cell. And so here, as a reminder, this is what 0% adapter dimer looks like in your bioanalyzer or tape station trace. Your peak here, no adapter dimer. These represent your low end and your high end markers. This is what 5% adapter dimer would look like. Again, here's your library peak, low end, high end markers. And just a reminder, as little as 5% adapter dimer contamination will lead to 60% of your reads at the end. And so again, you'll have to throw away 60% of your data if you have just 5% adapter dimers in your library. So super important to do QC on your libraries. Super important to do an extra cleanup to get rid of those adapter dimers if you are seeing them. Just reminding you of a picture of what your adapter dimer looked like. Again, there is none of your sequence cDNA of interest in here. So all your sequencing and adapter dimers is the adapters themselves. So next, we are going to go to the next step that happens after your libraries are completed, QC'd, and you have loaded the appropriate amount on your flow cell. The next thing that happens is once it's loaded on your flow cell, it's going to undergo cluster formation, followed by sequencing by synthesis. This is just a little cartoon that I took from Gold Bio of what cluster formation looks like. So here we have our template library, so either DNA or RNA libraries here. And then this also will include our sequencing priming region as well as our indices. And then this is showing just our P5 and P7 adapters in green and pink on either end of our inserts here. And so what you're looking at here is the flow cell. And then these oligos, again, A, T, Cs, and Gs, are going to be complementary to your P5 and P7 sequence that you added on to the either end of your adapters. And this here, this complementary P5 and P7 region is actually covalently bonded onto the flow cell, so very tightly bonded. And so what you're first going to do is when you introduce your libraries to the flow cell, these libraries are going to hybridize at their P5 or P7 regions to its complement on the flow cell, as you see here, right? However, at this point, our inserts aren't very strongly attached to our flow cell, right? Because it's just through these hydrogen bonds, which are pretty weak bonds. And so what happens next is your insert here that has your P5 and P7, where one of those sides are hybridized to its complement that's attached to the flow cell, is going to be used as a template, and we're going to add polymerase as well as DNTPs here to be able to synthesize a complementary strand of your libraries. And this time that strand is going to be covalently attached to the flow cell, right? So strongly attached, this copy is. And once we have that copy made that's nice and tightly attached to the flow cell, your original library is going to get washed away. You don't need it anymore. And now this nice covalently bonded insert that has your adapter regions is then going to fold over and covalent or in hybridize, right? So hydrogen bond to its, if that's P7, then this would be P5. And then once it is hybridized there, that acts as a priming region. So that way it'll recruit polymerase, which will make a copy in the other direction, right? So you'll have your forward copy or your reverse copy and your forward copies being made in this way. Once that is created, those will be denatured 2 strands, leaving you with 2 strands in each direction. This happens millions of times in each location on the flow cell. And so the reason that cluster formation is so important is because it is making millions of copies of each strand that you want to sequence. This is important because sequencing by synthesis is actually quite error prone. However, if you're sequencing the same strand millions of times, you are going to end up with the overwhelming signal coming from the correct base that's being incorporated into your sequence as you synthesize it. So again, cluster formation, making millions of copies of each strand that you're going to be sequencing. The reason for this is to minimize any errors that come from the sequencing by synthesis mechanism process. So at the end of cluster formation, you're going to have what looks like this is you're going to have millions of copies of each one of your sequence in these little clusters. So that's why it's called cluster formation, right? So we have little clusters of millions of copies of the same sequence that we're going to then go ahead and sequence with the sequencing by synthesis mechanism. And this image here is from the Illumina TrueSeq stranded reference guide. And so again, continuing with the Illumina method here, showing what it looks like for the sequencing step. So once we have our clusters, every strand in each one of those clusters is going to get sequenced the same way. And so our forward strand is going to be sequenced first, which means the reverse template is going to get cleaved off and washed away, leaving only our forward templates there. And so then it is going to utilize this priming region. The reagents that are added to the flow cell includes, of course, just like we saw in the pyro sequencing at the beginning of this lecture, the primer that's complementary to this priming region of our adapter that we added, and then a polymerase, which is going to go ahead and sequence this strand. And we're going to see a picture of what that looks like right here. So we are binding to our priming region, and then we are adding each one of our DNTPs that are attached to a different color fluorophore. And so your A, T's, your C's, and your G's will all be represented by a different color. And each one of those will have a blocker on it so that just one nucleotide can get incorporated per cycle. And so what happens is, depending on what the next base is in your unknown sequence, its complement is going to get incorporated. And then It's going to release a signal that's going to be captured in a camera. This block is going to be released that will allow the next nucleotide to be incorporated based on what its complement is in your unknown sequence. And we're going to take a picture after each cycle so that for every single strand in every single cluster, we're going to be sequencing and taking pictures after each cycle, so that it's going to look like this. And then based on the color that is emitted, we will be able to know if there was an A, T, a C, or a G in that location of your unknown sequence. Once this first read is synthesized and we capture that sequence in these images, the next thing that happens is that synthesized strand is going to get washed away. And then after it gets washed away, these priming regions are going to be, again, taking advantage of, but this time it's going to allow for us to sequence the index, right? So then we'll have the primer, we'll put on this complementary to this priming region, sequence in this direction in order to sequence this index. And then we would also do it on the other side if it was dual index libraries. So that way we have our forward read, and then we have both of our index sequences read. And then after sequencing, and then after that, sequencing of the reverse read is going to be initiated after your index reads. So how that happens is this forward template is going to fold over, bind, hybridize to its complement to create a copy in the reverse direction. And then this forward template is going to be washed away, allowing the reverse template to then be sequenced, which is going to be our read 2. So we'll see this here. Again, remember the reverse complement of this was made. So this is the reverse complement or our reverse template. And so our read 2, our reverse read, is going to be read in the opposite direction. In a couple of slides, I'm going to show you a link to a video from Illumina that I highly recommend you watch that's going to make this process hopefully a lot easier to understand once you see a little video of how it happens. So before we get to that, you set up your sequencer however you want. So you can set up your sequencer at whatever length you want up to the limitations of the flow cell that you're working with. So with Illumina flow cells, I believe the longest they have is a 600 cycle flow cell, which will allow you to sequence, if you're doing a paired end, 300 cycles on either side. I should also point out that although sequencing by synthesis works really well, we have not quite been able to replicate the conditions to make it as nearly as efficient as what happens in our actual cells, where in humans we are replicating with high fidelity 3 billion bases. So we're not quite that good. So longer reads, you will see the quality fall off a little bit towards the end of those reads, as we'll see in our QC of our sequencing data that we'll get into next. Also for sequencing depth, this will depend on how many samples you add to your flow cell and the number and the number of reads that you have from your flow cell. So again, just like Illumina offers flow cells with different number of cycles or different lengths, it also offers flow cells that'll give you different numbers of clusters, so different read depths. And so some standards that we aim for at GeneLab, so on the DNA-seq side of things, the longer your reads are, it enables greater confidence in things like taxonomic classifications and also functional annotations. And assembly is usually performed first when you have short reads in order to facilitate the taxonomic identification. And so we do want longer reads. So our standard at GeneLab is to sequence 2 by 250, giving us 500 roughly. Of course, we want some overlap, so 450 base pairs roughly is what we're looking for. In terms of sequencing depth, greater depth is going to increase the likelihood of sequencing low abundance organisms if you're doing this DNA sequencing for metagenomic samples, and also detecting things like single nucleotide variants, and genetic rearrangements with greater confidence. So some standards that we use at GeneLab is if you are sequencing DNA from a single organism or tissue, if you're doing resequencing, meaning that you have a reference to go off of, our standard is to do at least 10 times minimum the size of your reference, so 10 times coverage. If you're doing de novo sequencing, so you're sequencing an organism for which you do not have a reference, 50 times minimum coverage, right? So 50 times the size of what you expect your genome to be. And then for metagenomics, so this is where you're sequencing DNA from a mixed community, meaning you have multiple different organisms in there, recommending around, or our standard is around 10 million reads per sample at a minimum. And then you can go by there based on what your data looks like. And this, of course, you're going to want to sequence deeper if you know that your community has a lot more organisms. For RNA sequencing, for read length, again, longer reads increases gene ID confidence. And so the GeneLab standard is 2x150 for bulk RNA-seq. So that means you're sequencing 150 base pairs in either direction, so 300 total. So the template size that we're usually aiming for is around 250, 270 base pairs for sequencing. In terms of depth, greater depth is going to increase the likelihood of seeking low abundant transcripts, also detecting novel transcripts and quantifying isoforms. A good way to think about read depth is, let's say, for example, you have a jar of M&Ms composed mostly of blue and red M&Ms, and you have just a few, maybe 5% of your container is green M&Ms, right? If each one of those M&Ms represent sequences from different genes, your red M&Ms representing one gene, your blue M&Ms representing another, and your green M&Ms representing another gene. In order for you to detect that green M&M, right, that's not there in very high abundance, you're going to have to sequence a lot more, right? You're going to have to pick a lot more M&Ms from that jar before you hit a green one. So that's kind of the idea here. If you have a gene that's low abundance, the more you sequence, or the deeper you sequence, is what we refer to in bioinformatics, the more likely you're going to be to detect those low abundance transcripts. And so for us at GeneLab, our standard for mammalian samples that's prepared for with the ribodepletion method is around 60 million reads per sample. And samples prepared with the poly-A selection is around 40 million reads per sample. The reason those are different is because of the ribodepletion method, we're going to be left with several different types of RNA, right? Anything that's not ribosomal. Whereas with the poly-A selection method, we should have mostly messenger RNA in our samples, so we don't have as much of other types of RNA in there, so we don't have to sequence as deep to get what we want. And then always more biological replicates are going to be preferred over greater depth of fewer replicates. So just like in any experiment in biology, you really want to maximize the number of samples that you're looking at to increase your confidence in your results. And then for both DNA sequencing and RNA sequencing, paired end is preferred over single end. And this is because, again, the sequencing by synthesis mechanism is good, but it's not that efficient, and the efficiency tends to wear off the longer you sequence. And so sequencing in both directions for a shorter amount is better than sequencing in one direction for a longer amount, because you're just going to get higher quality data at the end of the day. And so this brings me to my last slide. Normally, if I was teaching this live, I would play this video. However, this video is not mine. This is a video from Illumina. It shows a great illustration of their high throughput sequencing by synthesis. I highly recommend that you watch this video, watch it multiple times. It'll really help understand how sequencing by synthesis happens on Illumina platforms. So again, can't stress this enough. Make sure you watch this video, watch it multiple times. Hopefully it'll put everything that we just went over with all together for you. This brings us to the end of our last of the 4 GL4U introduction lectures. I hope you guys learned quite a bit from these introduction lectures. including some overview of NASA, the Science Mission Directorate, space biology, the Open Science Data Repository, GeneLab. Also, hope you guys got a nice introduction to Unix commands as well as Jupyter. And then through here, I hope you guys have a little bit better idea now of how we prepare libraries for sequencing and how short read sequencing through sequencing by synthesis happens. At this time, after completing all 4 of these lectures, you are now ready to move on to the 4th and final Jupyter notebook of the hands-on content for the GL4U Introduction, which is where we are going to go through how to check the quality of the sequence data that comes off of a sequencer. The example we're going to go through is sequences from an Illumina sequencer, short read sequences. So go ahead now and transition over to that hands-on content and go through that 4th and final Jupyter notebook activity for the GL4U introduction. Thank you all.